Okay, well, welcome everybody, despite the weather. I appreciate those of you who could make it. February is half over. I always am glad that February is a short month. I don't know if people feel the same way. I'd like to remind you that this meeting is being live streamed on YouTube and that the recordings will be made available after the fact. We appreciate your understanding of that. Again, introduce myself, James Glazier, co-lead of the working group with Reinhard Laubenbacher. We have support from Jim Sluka, who's done web administration, a lot of scientific content as well, and then also Bruce Shapiro, who you hear from on a regular basis. If you want to contact us, we're always available to respond. Also remind you that we have a Slack. IMAGMSF is going to be pushing Slack more. We had a message from Grace saying that she wants all of IMAGMSF to be using Slack, not just us. And so again, I please encourage you to use it. And if you need access and have trouble, let us know. Also, the IMAGMSF wiki pages always need content and love, and your assistance with that would be much appreciated. I want to remind people again, next week on Monday and Tuesday, there is a workshop being held by NSF. NSF is planning some large-scale center-type RFAs on the topic of preventing the next pandemic. They're going to be interested in experiment and modeling of various kinds. The workshop next week is, I believe, mostly going to be focused at molecular and population levels, but they have Denise Kirschner speaking, doing multiscale modeling, a variety of interesting speakers. The organizers I talked to yesterday are eager for overlap and connection with this IMAGMSF group. You can still register to hear the talks and participate. Prevent-symposium.org, and I think it's a good opportunity. The focus comes from rather different background. In-host modeling, I think, was not primarily their focus, but they're very interested in the connections with what we're doing. And so I would encourage people to check it out. It's going to be essentially from 10 a.m. to 5 p.m. Monday and Tuesday of next week. A reminder for upcoming meetings and mini-seminars. Tomorrow morning at 11 a.m. we have our steering committee meeting for this organization. Hope I'll see many of you there tomorrow. February 25th, we have two great speakers lined up. Rita de Almeida from Brazil is going to be talking about a very interesting bioinformatic way of aggregating expression data to provide quantitative information for model validation. And Jim Fetter from Pitt is going to be talking about some of his elegant work on modeling viral replication. March 4th, we have Yi Zhang from Georgia State on the books. We have slot room for another speaker, and we're always looking for additional speakers. So please either volunteer yourself or suggest people you'd like to hear. We would really appreciate that. Now, we've been having a good time with these meetings, I think. Time always is too short. And so to try to make sure that these are maximally effective, I would say that one thing we would like is to hear, of course, to quote Yogi Berra, if people don't want to go to the ball game, nobody can stop them. So if you don't want to ask questions, you don't have to. But we have wonderfully diverse expertise in the room. And so we'd love to hear some questions from the people who have been listening so politely and silently over the past few months. And so we really would encourage people to ask questions. especially the people who have not been asking questions. But those of us like me who ask questions all the time, maybe we can hold back and let some of our more reticent colleagues have a chance. So I'd encourage us to allow that. And I also would say that because time is so limited, if we could focus on more general questions, we typically only have time for 1 or 2 questions in this main session. And then what I was proposing, and I'd sort of like a show of hands perhaps as to whether you think this is a good idea, it will depend to some extent on our speakers, would be we have a very tight hour cutoff for this meeting, but we could create a set of breakout rooms for informal discussion for a half hour after the end of the formal meeting. And that would allow the speaker and people who are interested to talk freely among each other, ask more detailed questions, have getting to know you opportunities, sort of meet and greet. I would create 2 breakout rooms, and then after the formal meeting, I'd let people move into those breakout rooms as they request, and you'd have a half an hour. Does that sound like a good idea to people? We couldn't implement that this week because we didn't get permission in advance from our speakers, but we'd like to start that next week. And then you could have your opportunity for technical questions then. And then the other thing is it's hard to keep track of how long you're speaking. And so I'm going to give people a 5-minute warning. Just if you hear me break it and say 5 minutes during your talk, I'll do that, try to do that systematically to stay on schedule. Okay. So with no more ado, I'd like to turn over the floor to Tomas Helikar from University of Nebraska, Lincoln. We actually heard some of his mention of his work a few weeks ago. And that was a nice teaser, and I'm looking forward to hearing his talk now. Tomas.
Thank you, James. Let me just share my screen real fast. Do you see my full screen presentation mode? Okay, thank you. So thanks again for having me here today. I will talk about one of the aspects of our research program, and we're at the University of Nebraska. I'm in the Department of Biochemistry. I am a computational biologist by training, and so being housed in the Department of Biochemistry has been very interesting in a very good way, in that I've had the opportunity to work with experimental biochemists who are dealing day to day with the biological processes we're modeling here. And also it's been giving me an opportunity to slowly but surely convert experimentalists into computational modeling, or at least start dipping their toes in there. And so today I'll talk about Cell Collective, which is a computational modeling platform that we've been developing since, I guess our first paper on that was back in 2012. So for quite a bit of time. And there are a couple of reasons and sort of core principles and goals that we have set out when we decided to start building Cell Collective. And one of the most important ones is to really make computational modeling accessible to non-computational biologists. It dawned on me, or I realized that back in 2008, 9, when I was still finishing up my PhD and working with a cancer biologist, and we were building a model of a computational model of signal transduction in cancer cells to try to understand the different pathways and potential drug targets and things like that. And it became very clear that trying to communicate our modeling results with experimentalists who are not familiar with computational models, but they also may not care about the nuts and bolts of a computational model, the mathematics behind it. It dawned on me that really we need a better way to communicate models and to make models more accessible to beyond computational, the modeling community. And in doing so, it also creates the opportunity for developing these models in a more collaborative and scalable fashion. Going back to my experience again, we built this model that had hundreds of components and thousands of interactions. And I read, during my PhD, I read about 700 primary papers to put that model together. And it, again, it became very clear that if we want to get to a point where we have very comprehensive, detailed, and accurate models, there is probably not a single group that can do it alone in a reasonable amount of time. And so there is a need for collaboration if we want to make the models bigger. And we also need the models to be more transparent through annotations and providing evidence as to why a particular interaction should exist in the model or not, or why it exists in the model. And I believe that will also further give rise and facilitate reusability and reproducibility of these computational models. And so Cell Collective is a web-based platform that you can find at cellcollective.org. And over time, it's evolved. It started as a modeling technology for scientists to model different biological systems. But over the last 5 years, we've also found that, or realize that you know, when we think about how we as scientists study biological systems through models, and then look at how we teach our biology and life sciences students about the same biological processes by having them memorize static pathways in textbooks, there is a big disconnect. And so we have expanded Cell Collective to also be a fully research or scientifically authentic educational platform. And so when you go to the landing page, you can choose to go to the research user interface or to the educational side of the interface. Today I'll be talking about the research side, and if James and Reinhard don't get tired of me, maybe they'll invite me back to talk about the education application of Cell Collective sometime in the future. So Cell Collective started as a logical modeling platform, and so probably many of you are familiar with logical or Boolean models. So I won't go into much of the detail about this, but there are some modifications that we've made to this mathematical framework that can be experienced in Cell Collective. And so in logical models, in Boolean models, every component is connected with the directed edge, and then the regulatory mechanism of that component is described with a Boolean function. So in this case, in time t+1, the component C will be active if A or B are active in time t. In Cell Collective, it is a Boolean network at any given time point, and you can see the real-time simulation. But you can also see that the output and input actually can be sort of semi-quantitative. And so what we have done is that we treat the external components as probabilities of that component being active or inactive in any time point. And so it becomes a Markov process, but you can still, the output can also still be completely binary, or it can be, as you will see in the next right here, you can look at the activity level in terms of a proportion of zeros and ones over a certain sliding window. So then you get a moving average, and that allows us to actually simulate more semi-quantitatively the impact of a perturbation on the different components of the network, or the impact of changes of dosages or activity levels of the external components. And so, as I mentioned before, making the technology more accessible has been one of the first sort of objectives that we set out to do. And so you can easily build these models basically by drawing a network, and it's building the Boolean logic all in the background. Of course, more complicated regulatory mechanisms can be defined by dragging and dropping components into as positive regulators or developing conditions and subconditions, and the tool will construct all the Boolean logic in the background. Most recently, a couple of months ago, we actually expanded Cell Collective to also support genome-scale metabolic models using constraint-based modeling approaches, and that created new challenges. And one of them, in terms of making navigating through the models, you can imagine that genome-scale metabolic models are going to be much more complex than the current logical models that have been constructed and have been available. And so making the visualization and navigation through the web browser is very difficult. And so we've developed a new JavaScript library, WebGL library, that allows you to actually very easily and nicely, without any performance issues, navigate through very complex networks. In terms of annotations, every model in Cell Collective has its own knowledge base workspace, where you have the opportunity to annotate and also review the level of annotation for every component of the model. And so you can annotate the components at the component level. You annotate the regulatory mechanism. Every single interaction can be annotated with pieces of evidence as to why that interaction exists in the model. These little icons, by typing in a PMID or DOI, you can also go to the level of detail and indicate whether that publication is a primary source or a review article, whether it comes from human data or animal models, and so forth. And so here, actually, this model here, this network, the colors, the green color corresponds to interactions that have been annotated. If you had red or orange colors, then that would be telling you that your interactions are not very well annotated, and so gives the user a sort of higher view of the level of annotations of the models. Again, trying to make it as easy as possible for modelers to understand the extent to which they have been annotating their models. There are several different simulation tools in Cell Collective. Here is one that shows a real-time simulation, and again, trying to make modeling as accessible as possible. We provide a bunch of dials that you can increase the level of activity for the external components. You can easily select the components that you want to perturb, for example, to simulate a gain of function or loss of function feature. And then you can see the activity levels of the whole system here, or a time series graph as the simulation evolves. And you can change that sliding window that I mentioned earlier here, that gives you either, if you set the sliding window to 1, it's a purely Boolean output. If you increase it, then you get more semi-quantitative output. The fact that the external inputs are probabilities or sort of intensities of that component, of that external component, gives us the opportunity to look at input output or dose response type analyses, even with Boolean models. And so you can run hundreds of different experiments looking at the impact of external inputs on different components of the system. You can also do environmental analyses, where you can ask a question: what would happen? So the model that you've been seeing is a bacterial chemotaxis model, a very simple system. So in this case, you can ask a question, for example, what environmental condition would maximize the running motion of that system? And so So it will run, again, you can base it on as many simulations. So this is simulation driven, experimentally driven. And so you run 1,000 simulations under different conditions of external inputs, and then it'll optimize, it'll find the most optimal conditions under which you will achieve that, objective. And you can have an objective that's more complicated, where you want to maximize one output, but you want to minimize the other output, and so forth. Another aspect that I had mentioned before in Cell Collective is to make it enable more collaboration, and also make access to the models more easy. And so any model in Cell Collective you can export as a SBML or SBML qual for the logical models. You can also share your models directly with collaborators, much like Google Docs works. And you can also make the models available through an anonymous link. So what we've been doing is that when we submit a manuscript for publication, then we can generate this anonymous link, give it to reviewers, who then don't have to create an account in Cell Collective, and they can review the model as part of the manuscript review process without jeopardizing their anonymous status. Then to publish the models, we have a criteria that if you want to publish the model, you actually have to have every component annotated. And so I realize that maybe that will prevent some people from wanting to publish their models, but I think I would put the preference on publishing annotated models more than having more models published and less annotated. And you can also publish the experiments associated with that particular model that you're publishing. Cell Collective, as I mentioned, because of SBML qual and SBML that also works for constraint-based models, has the ability to talk to other software tools. And so if of course Cell Collective doesn't have all the features that other tools may have, and so through SBML it's very easy to build models in Cell Collective, do some analyses in there, but then also export the models and make further explore those models using other tools. So currently Cell Collective has a little over 200 models. We're still in the process of integrating models from BioModels and BiGG Models in terms of the constraint-based models. Of these 204, there are 90 logical models that we have manually curated and rebuilt from published literature, because many logical models had been published before SBML qual even existed. And so we went back to the literature, reconstructed these models in Cell Collective, curated them such that they are equivalent to the published models, or models available in those published papers. We back annotated them because, again, as you will see, and probably even from your own experiences, you'll see that models in published models have varying degrees of annotations and supporting evidence in those papers. And so so we went back and re-annotated all the models to make it more consistent. There are a lot of different types of models. I won't talk about the COVID, but Cell Collective, Marek and Anna had already talked about the project a couple of weeks ago, I believe, about the international effort to map COVID-19. And so Cell Collective, among many other tools, has been providing the support and infrastructure for the models that the community is building. What I will talk about— 5 minutes? Okay, thank you. I will just run through a couple of other examples that are relevant to this group. And so in this particular case, this paper is back from 2012, but we have recently resumed working on host-pathogen interactions, where we use Cell Collective and our modeling approach to model the replication cycle of influenza. And so many probably know that influenza gets internalized into airway epithelial cells. The genome content gets imported into the nucleus, and then the virus hijacks the replication machinery of the host cell to produce progeny virions. And so this is relatively simple but complex or intricate process, and so we mapped it out into a logical model, and we focused on the receptor tyrosine kinase pathway that is critical for the virus to enter the cell, and looking at comparing different ways of inhibiting or intervening with the replication of the virus. We were able to we compared these different inhibitors at that time, PB1 and PA interface inhibitor, which is part of the viral replication component. That interface inhibitor was just being explored experimentally, but you can see by being able to put the external inputs on something other than just zeros and ones, you can actually look at potential dose response curves related to inhibitors and their effect on the different components of the system. Most recently, we've resumed working on this project, and we have actually built out a much more comprehensive model of signal transduction in epithelial cells, and we have been able to identify drug targets that could target the host side of signal transduction that we're currently validating in vitro. Another project that is very recent, we have a preprint out on that, is to model the immune system as a whole at the cellular level, and so including both adaptive and innate immunity. We created this logical model that contains 10 different pathogens and 164 different components, including 35 different cytokines and nearly 30 different immune cell types. So what that now allows us to do is to look at the characteristics of different infections, and also the different characteristics of potentially different co-infections that are many times common. So to conclude, so right now our long-term goal, so Cell Collective right now supports the modeling of logical models, so signal transduction and gene regulation, but also now genome-wide metabolism using constraint-based models. We're beginning our efforts to include support for differential equation-based models, and then ultimately being able to start connecting these into multiscale and multicellular system. And again, to kind of drive home the message, our goal of Cell Collective is to really make modeling a communication tool among the different types of scientists, ranging from basic science experimentalists to computational biologists and to translational scientists. And so I will end with that. Thank you very much.
Thank you very much. That was a lovely presentation, and I think there'll be a lot of interest. In the interest of making sure we get through the second talk, may I ask that people hold their questions until we've finished the second presentation today? Is that okay, Tomas?
Yeah, absolutely.
Okay. Yep. I just want to make sure that we don't run out of time. Usually we have time, but okay. Thank you again.